Dr. Aoki is the head of the Department of Molecular Therapy and an adjunct professor at several prestigious universities in Japan. As the Japan Rare Disease Consortium leader, Dr. Aoki is committed to fostering innovation and collaboration to overcome neuromuscular diseases. Dr. Satya Prakash, who is joining us tonight, has been a postdoctoral research fellow at the National Center of Neurology and Psychiatry since 2021, working on setting up human 3D model brain organoids to model cognitive phenotypes in Duchenne muscular dystrophy. And tonight she will give us an update on her and Dr. Aoki's current research into iPSC-derived brain organoids as a model for cognitive phenotypes of Duchenne muscular dystrophy. Welcome, and thank you so much for being here to give us this update. I really appreciate it.
Hi, thanks so much for the intro. It's actually Friday morning here, so
good morning. I'm so sorry for forgetting
so hi
this.
from Japan. No worries at all. I'm just trying to share my slides. Is that the full screen?
I think we're seeing the speaker view.
Oh, okay. Is that better?
There we go. Yep, that's great.
Okay,
Thank
great.
you.
So thank you so much. It's a pleasure to join for the 30th anniversary. So I'm just going to give an update on, it's actually the first year of the funding that's of a 3-year research project. So our project, of course, we're looking at DMD, but just as a kind of sidestep from the muscle work we've been hearing about, I wanted to highlight that we're interested in studying the molecular pathways that occur as loss of dystrophin at the brain. And the brain expresses not just the full-length dystrophin, which we see in the muscle, but also several shorter phenotypes, shorter isoforms, such as these listed here. And we're specifically, for this presentation, focusing on the cerebral cortex. Now, DMD has lots of cognitive disorders associated with it in around 30% of patients. But unlike the muscle disease, we don't clearly understand why loss of dystrophin leads to these various spectrum of disorders in the brain. We do know that these isoforms, particularly this DP140 isoform, are expressed during development. And the pattern of the expression of these proteins changes from before birth to after birth. So clearly there's a very specific function that we need to isolate. But of course, that's very challenging. You can't just directly look at fetal human brain. So instead, we've developed this protocol from induced pluripotent stem cells derived from the urine-derived cells directly from patients to make cerebral brain organoids. Now, this is a protocol where you take induced pluripotent stem cells, which still maintain the genetics of the patients, so they still have the various DMD mutations, and then we can grow them by exposing them to different factors in kind of growth media in a petri dish to kind of recapitulate specific regions of the brain. And of course, these little brains are going to be deficient for dystrophin. So we can look at the effects that this has on specific time points of development, just like kind of specific growth points during embryonic development. And so we were able to do that. We've got 2 individuals with no DMD, and then 3 different patients with different mutations. And from here, we were able to grow cerebral organoids. So aim 1, year 1 of this project, was to try to characterize the spatiotemporal expression of dystrophin isoforms in the cerebral cortex, as well as other types of organoids. But for now, we're focusing on cerebral cortex. So we did this at mRNA level, and essentially we found that brain-specific isoforms were downregulated, as you might expect, but they weren't totally lost. So this is the key takeaway, that we see a kind of misregulation of dystrophin isoforms in the brain, but not total loss. And when we look at the protein level, we're able to capture not just the different neurons that are expressed in brain organoids, but also specific regions. So during brain development, you have neural progenitors, which then differentiate into neurons. And we found that dystrophin is expressed not just in the neurons, like we've originally thought for several years, but also in neural progenitor regions. And this could have significant implications for when in DMD organoids, we lose these brain specific isoforms. Now, this work is currently being analyzed, and we're performing several differentiations of brain organoids to clarify exactly what effect that the loss of this dystrophin is having on our brain organoids. So I just want to say that overall, we've been doing lots of experiments with the brain organoids, but our year one goals were pretty much mostly achieved. We just have a couple of modifications, which I'll come back to in my final slide. But we've been able to not just generate several brain organoids and look at the dystrophin expression at mRNA level, but also at protein level. We've also been able to move on and done some preliminary experiments showing the morphology of our brain organoids, specifically cortical markers, specific brain markers, which are really important for development. And we're going on to perform more of these experiments in hippocampal and ganglionic eminence organoids, which should be really crucial for the phenotypes that we see in DMD. So the final experiment, and this is really crucial because without this Defeat Duchenne Canada funding, we definitely would not have been able to move on. We're currently funded by an additional grant from here in Japan, but thanks to this grant, we're able to kind of upgrade one of our experiments of in situ hybridization to look at spatial transcriptomics instead. Now, this is a really novel next generation technique where we'll be able to look at specific isoforms of DMD expressed at different time points and regions of the brain organoids. And this will pair really nicely with single cell RNA sequencing experiment, which we have planned for year 3 of the grant. So I'll be really excited to share these updates with you in the following year. So I'll just end by thanking everybody who's been involved in the group, especially the patients as well who've donated their cells for this project. So thanks so much.
Thank you so much for that update. It's fascinating. I appreciate your taking the time out of your, I'm sure, busy morning. Thank you so much.